wondered what it would be like if we could cure genetic disorders with an IV, even before they present themselves. Okay, that sounds like a stretch, I know. But as it turns out, we might be getting closer to this than you might think. You're probably wondering how. Well, gene therapy. This is something that could change the game for modern medicine. It involves the insertion of a functioning gene into cells to compensate for an abnormal gene or to make a beneficial protein. It uses a normal copy of the gene to restore its original function. It may sound a little bit complicated right now, but picture it like this.
Let's say this puzzle is your genetic code. Each piece is paired with a specific other. Their shape allows them to fit together into the whole puzzle. But in this puzzle, there's a problem. A piece doesn't fit. This piece alone can cause the whole puzzle to become distorted. This would be called a genetic disorder. And that's where gene therapy comes in. It basically swaps the damaged gene and replaces it with a normal and healthy one.
Gene therapy, while it may sound new and even futuristic, has actually been around since 1963. Of course, back then it was merely an idea, as it could not become possible with the development of recombinant DNA.
Recombinant DNA is the joining together of DNA molecules from two different sources that are then inserted into a host organism to produce new genetic combinations. After it is created, there is a need for something to transport it to the nucleus, which is where the chromosomes and the DNA are located.
This is where vectors come in. Vectors are basically like molecular delivery trucks. There are two main types of vectors, viral vectors and non-viral vectors. Viral vectors are the most commonly used vectors since they are extremely efficient at transferring genes. And gene transfer through viral vectors is referred to as transduction.
Imagine these trucks are the viral vectors, and that's your cell. They are especially good at introducing their genetic material into the host cell as part of their replication cycle. It's second nature to them, it's what makes them such good vectors. To use a virus as a vector, first you would have to remove the genes that cause disease. That would be the gravel. And then replace them with the desired ones. That would be the sand. The genes that allow the virus to insert its genome into the host must be left intact. The concept is pretty straightforward, but actually very hard to execute because vectors can pose a threat too. For one, non-viral vectors are very ineffective and viral vectors can cause some problems. For example, since viruses can affect more than one type of cell, when used as vectors, they might infect healthy cells and even cancer cells. Another danger is that the new gene might be inserted into the wrong location of the DNA, and that can cause more mutations and even cancer.
So yes, it's not as simple as it first appears, but even though scientists are still figuring out the details, gene therapy shows much promise. It is just the beginning of a whole new world in medicine, and it could revolutionize the way we treat genetic disorders and actually help us find a way to cure them. It may seem far-fetched and a long way in the future, but maybe that future isn't quite as far away as you think.